Michelle here, and today we're going to learn how to draw a typical eukaryotic cell using a human liver cell as an example, and that it's actually part of the syllabus in Unit 1.2 in IB Biology. So it is a topic that's covered in both standard level and higher level. So if you're interested to learn how to draw a eukaryotic cell, then yeah, grab a piece of paper right now, grab a pen, pencil, okay, and let's just draw this cell together, okay? So of course, if you do know how to draw this cell already, you can skip to the very end of this video and you can see and check if yours is similar to mine, or is there anything that you can modify. Yeah, so anyway, let's just get started. So here, first thing first is when I'm drawing a eukaryotic cell, I would draw like a huge circle first. Okay, so mine is definitely not a perfect circle, but yeah, I think it's good enough. There's no cell that's perfectly, in the circle shape anyway. Okay, anyway, so moving on, one of the main difference between a prokaryotic cell and eukaryotic cell is definitely that eukaryotic cells do have nucleus, so that's why we need to draw a nucleus. So for nucleus, because nuclear membrane has pores, so that's why I would usually draw. So it's up to you. You can actually draw like another circle inside first, and then you can use like a little eraser, and then you can just clean up like parts of the circle like this, so it kind of creates this kind of nucleus with nuclear pores sort of effect. And then we would draw like a smaller circle inside, and I usually shade it in to kind of demonstrate like the difference between that's the nucleolus. And then I would draw some lines inside that represent the chromatin or any genetic, the DNA that was stored in the nucleus. Okay, so moving on, usually I would tilt the paper a little bit just for me to draw this easily. So I'm gonna draw some of these little pancakes. I like to call these pancakes. I tend to call a lot of different things pancakes in bio. I don't know why. I call those a thylakoid and a chloroplast pancakes. So yeah, I guess I just want pancakes right now. Anyway, so you have to draw like similar structure like that all around a nucleus, like so. You prefer, you can also draw some on this side, but if you don't want to, and if time is running short, you definitely don't have to draw all of these. And so these basically are rough endoplasmic reticulum. Rough endoplasmic reticulum won't be considered rough without all the dots on top of it. So these obviously will be the ribosome. So make sure when you're labeling this later, which we'll do very shortly, specify on the type, the size of the ribosome, okay, on these rough endoplasmic reticulum. So after drawing the rough endoplasmic reticulum, of course, we do need to draw some smooth endoplasmic reticulum. So those that I'll use to synthesize lipid, which they don't really have any ribosome on top of it. So just add a little bit more, okay, nothing to excess. All right, so moving on, we're just gonna draw some similar structure on the outside. Okay, on the outside of the rough endoplasmic reticulum, nothing too crazy, but yeah, just a little bit more here and there. And then we can just label that as smooth endoplasmic reticulum later. All right, so that's pretty much it near the nucleus. And then we can move a little bit further away from the nucleus towards the plasma membrane. And of course, we need a machinery to modify and package protein, so polypeptide. So of course, we do need to draw. Why am I on this? Okay, so we'll have to draw some of these sort of, it looks kind of like a Spotify logo, but not exactly. Okay, so we just have to draw something like that and some circles next to it. Okay, that would basically represent the vesicle that's packaging all of these protein that is going to be transported out of the cell. Okay, extracellular proteins such as like proteases. Why did I mention proteases so specifically? So it could be enzymes, it could be hormones, it could be neurotransmitters, okay, or maybe some antibodies. Okay, so moving on, we do need to draw mitochondria, so don't forget that. So make sure when you're drawing a mitochondria, you kind of want to emphasize on the fact that they are like double membrane, and the inner membrane is sort of like yeah, like highly folded to form this like crystal finger like projections inside. Okay, so moving on, let's not forget to add more dots in the cytoplasm, and that are these free ribosomes that makes protein used inside of the cell, intracellular protein. Okay, so of course I would also draw some like smaller, another circle that has some more dots in it. And those are the lysosome in the cell that contains digestive enzymes that kind of breaks down unwanted material inside the cell as well. So that, and then if I were you, I usually would add some structures like that too. And these kind of really cute looking, almost like a magic little wand kind of structures. They are centriole. Okay, you can pause this video right now and think about, well, a centriole, what's the function of centriole? So if you remember, centrioles, okay, I hope that you paused it, but if you did, that is fine. Centrioles are structures that are mainly used for when a cell is dividing. So it forms a spindle fiber that attaches centromere to chromosome, and then it just pulls. Yeah, very important. So that's that. And then let me see, is there any other structures that we need to draw? I mean, pretty much that's really about it. We could start labeling, and if there's anything that we need to add, then we just add it later. So make sure when you're drawing this in exam, use a pencil, okay? And make sure you just stick with pencil even for the labeling. But then right now, since I want to demonstrate and make sure that it's easier for you guys to see, I'm gonna use like a red pen, okay? Red color. So we start with plasma membrane, cytoplasm, Golgi apparatus. Label these are vesicles. And then we have ATS ribosomes. Mitochondria. So yeah, also one thing, I would strongly suggest you to use label lines instead of using like an arrow, because it's just easier to see what you're labeling, and it is more kind of, it can really point to what you actually want to label, all right? So yeah, that. And then we have the whole thing here. So this whole thing, you can just label this as the rough endoplasmic reticulum. And then here we have the nucleolus. Here we can just label DNA or chromatin. And then we have to label these as smooth endoplasmic reticulum. Smooth, ooh. And then, of course, this whole thing here, you can just label this as the nucleus. And yeah, that's really pretty much it. So yeah. Yes. So, okay. This is how we actually draw a eukaryotic cell. So we're all done. Okay. So if you have any questions, please leave it down in the comment section below. And good luck with revision in IB biology and other biology. I just love biology. So till next time, see you then. Bye-bye.